Right, ladies and gentlemen, welcome to the Oxford Café Scientifique. For those online or in the room who are new, just to remind you that this is a short talk, long discussion format, very informal. If you want to go and get more snacks and drinks, or if you need to go and catch an early bus, we are very forgiving. We will take a break after the main talk. Please, can anybody online put any questions or comments into the chat? And I will then read them out for you. If we really can't understand it, we might ask you to join in, but it's a little difficult to get the audio to everybody in the room, so we do tend to do the online version from the chat. Bit of housekeeping. This is the beautiful Pharmacology building. Should there be a fire alarm, which we're not expecting that to sound, that would not be a drill, then we have to leave because we do have some potentially flammable things in the building. Two ways out. You can go down the main stairs, the lifts will stick, so don't try those. We can go down the back stairs if you're in the room, you can see there, that will take you out to the back of the building. If people can make their way around to the front of the building, over by the fence next to the porta cabins, that would be perfect, and I can do a head count there. Just make sure we've got everybody. So that's fantastic. If we have to do that, obviously it will go dark because I will be pulling the plug on the recording. That's basically it. The way that this works is we have a short talk and a long discussion. It is designed to be for anybody with any scientific knowledge or none. There's no such thing as a silly question is the motto of this cafe. So that's the kind of background for it. As you'll see, if you have a bit of a look at our website, if you use Oxford Café Scientifique on Google, you'll find it very quickly. The website's now back in action, updated. You'll see we've got a load of talks already in, and a whole other half dozen speakers trying to decide which day to come. So they'll fill in more and more as time goes on. And if you're not on the mailing list, you can add yourself, and that would be great, and then you'll get notification. I do record these, so this is recording at the moment through Zoom, audio and video, and then I will do a whole upload fairly soon. Probably this one will go up quicker than normal because it will go up with lots of others. If you really, really don't want to have your voice...
I need something to stop it moving.
Okay.
Can you turn your audio off, please, online? I'll mute them in a minute. I don't have a helper today. So if you really don't want to be heard asking a question, then it's fine. I'll ask it for you. Just catch me. We have suggestion slips and bits for making notes on the table as well. So I'm absolutely delighted tonight to introduce Dr. Kathy Potter. Kathy is Canadian by birth and training. So she did her degree and then doctorate over in Canada, firstly at Montreal and then in Kingston, Ontario. She had a very long and varied postdoc career, moving around from Canada to Idaho and then Toronto and Edmonton again, and then migrated over here to the UK, where she spent a fair chunk of her career, and has recently retired as the manager, from being the manager of the tissue bank, human tissue bank in Southampton University. But is also a lecturer in immunology still, and this is her pet topic for discussion. And thank you very much to the audience member who put me on to suggesting her. Because it's fantastic. We've had a great chat, so I know she's a good speaker. Over to you, Kathy.
No. Thank you very much for that wonderful introduction.
You're on one of the blank spots. Move back a bit. That's it. Perfect.
Got it.
Perfect. You can go either side or
just down the center.
It was just that bit by the table, which is a problem. Okay.
So we're here this evening to have a discussion on the role of jumping genes as co-authors in the DNA books of life. And I put books plural, meaning that I'm not going to just be talking about human genetics and genomes. And then there is the concept of life, whatever that is. In 1870, Paul Ehrlich said that it was a chemical incident. And that pretty well captures it. Oops, what am I doing? So here's the modern human genome. Let's see if I can go into it. So we've got 23 pairs of chromosomes, 46 chromosomes in total. And here's a staining pattern. Here we've got XY for male, XX for female, and it took a lot of work to nail this picture down. And hence nobody wanted it messed up with the concept of jumping genes, because that means that a segment of DNA could pop out of one place and elbow its way into another place. And you spend all this time nailing this picture down, that when the concept of jumping genes was first brought up by Barbara McClintock in the 1940s, the general reaction of the scientific community was, no way. Okay. So are the genomes static or dynamic? What would happen if your genome, your chromosome was broken and DNA from another source, or even from another part of your own chromosome, went in there? Can genes move around in genomes? Can genes be lost? Can external genes be inserted? These are the topics that we're going to discuss tonight. So the kernel of an idea first started with Barbara McClintock. Yeah, in the 1940s, 1950s. She was studying corn. So this was maize. And she was using inbred maize. So from this ear of corn, she knew who the parents were, the grandparents, and the great-grandparents. It was an inbred ear of corn. The expectation was that all of these kernels were going to be colorless. And you don't need a microscope to see that they're not. And some of them, in fact, are making tremendous amounts of pigment. And so, as a scientist, she said, you know, what's going on here? And she set about to work analyzing all the chromosomes in all the individual kernels of corn. And she provided the evidence that the genes can move from one chromosome to another under the control of the organism. And she and everybody accepted them as being called transposable elements, or more colloquially, jumping genes. But the scientific community at the time in the 1930s just said, no way, can't be. This would just be genetic chaos. And how would you have, you know, children looking like their parents if genes were fluid and moving all over the place? 30 years later, she won the 1983 Nobel Prize for the discovery of jumping genes. So something happened in that 30-year time frame that got the thinking of the scientific community to move from being a dead, no, your results have to be wrong, to yep, jumping genes exist. And whoops, just a second. Okay, just to make sure that we're on the same page, I just wanted to go over the cell, because this is where all of our DNA, all our chromosomes are located, is in the nucleus of the cell. We humans have a cell that has a membrane around it, so it's called a eukaryotic cell. Here's our DNA. That's the good old double helix that you see pictures of it all over the place now. Okay, and it's tightly wound up on proteins and compactly packaged into a chromosome. And then the chromosomes that I just showed you the picture of are all kept safely in the vault of the nucleus. Okay. Now, the cell, our human cells, each cell is actually functioning as a miniature computer. And it's sending WhatsApp messages from the nucleus to the endoplasmic reticulum. And they're decoded in the endoplasmic reticulum in order to make a protein. And then the protein functions to keep either that cell alive, or it can get secreted from the cell and go function, you know, at a distance and an organ quite separate from the cell it was manufactured in. So the surface of our cells are not like billiard balls, they're more like tennis balls, meaning that they're furry. And what we have is we've got a lot of antennae out there on the surface of the cell that are sampling the environment. And when it gets hit with something, it picks up on something, this protein will send a signal through to command central, which is the nucleus where the chromosomes are. And all the different messages coming in from all the different sensory elements end up at the nucleus, and the nucleus has to decide what it's going to do. Is it going to just sit there and stay alive, but not actually do anything? Or is it going to die because there's programmed cell death? Is it going to proliferate, make more of itself, double everything and split, double everything and split? Or is it going to differentiate? And that means a change of personality. And we're going to go through examples of this, is where a cell maybe could be a jack of all trades and then decides to just be a protein manufacturing site and specialize. Now, this is where all these 46 chromosomes are located. There's another source of DNA in our cell, and that is the mitochondria. There's DNA in the mitochondria. The mitochondria is thought to be a very ancient bacteria that got into a cell of one of our early ancestors, took up residence, and then for a safe rent-free existence said, I will make the energy if you let me stay. And so the cell and the mitochondria came up with an agreement, and that's why we have mitochondria to this day. Okay. However, what we can see is that we can have like several thousand of these double-stranded circles DNA. Each one of them is 16,659 bases. Now that's kind of small compared to our chromosome, which has roughly 3 billion base pairs. That makes 20,000 genes. And this is all filled with encrypted instructions. Okay. Oops. So just to make sure we're all on the same wavelength here about DNA, double-stranded DNA, we've got the rungs of the ladder, and each strand is composed of a nucleotide, which has 3 components to it: a base, a sugar, and a phosphate. The base is either A, T, G, or C, and that's what I mean by the Book of Life is written with a 4-letter alphabet. English is 26. The Book of Life, and if you think of variety in life, 4-letter alphabet, okay? There's going to be a lot of grammar and punctuation involved and jumping genes. So just to make sure you know what we're talking about is that in order for this double strand to neatly fit and also be a helix, A pairs with T and G pairs with C. So it's a big one and a small one, a big one and a small one. Because if you get the 2 small ones, it sags, and if you get 2 big ones, it bulges. So A pairs with T, G pairs with C. It's called complementarity. Okay, so, and the only, the sugar here, here's the base, here's the sugar, here's the phosphate. Base, sugar, phosphate, base, sugar, phosphate, base, sugar, phosphate. The sugar here is deoxyribose. Deoxy, D means minus, And what is it? Minus, it's minus an oxygen. The sugar ribose has an oxygen here, and in DNA, it's missing that oxygen, so it's called deoxyribose. That's why we get the name DNA. So what happens is that you've got a strand that it's got information in it, it's encrypted. You want to make a protein with amino acids in a certain order. That's the object of the game here. However, the DNA wants to stay safe in the nucleus. It doesn't want to go out into the wilds of the cytoplasm and into the ER, where there could be enzymes and all kinds of business. So what happens is that it gets a scribe, which is the polymerase, to write the message in RNA, and it's quite free to send the RNA out to the endoplasmic reticulum. And it's a little bit like in older stuff, particularly when you had the negative to make the positive. And so this is, the polymerase uses the negative to make the positive, because this is what's got the information in it. And this is what gets decoded. Now, yeah, so this is decoding the nature's genetic code here. We've got 20 amino acids. We've got 61 sense codes. We've got 3 stop codons here. And so this is where we're getting punctuation and grammar. And so we've got 64 triplets of bases that make a series of amino acids. Now, this is what happens when the DNA made the RNA. This is the messenger RNA that went out into the endoplasmic reticulum. What happened is that a ribosome jumped onto it. It comes in 2 pieces, a large subunit and a small subunit. It jumps on. It's got a cavity in it. And in that cavity, you get 3 of the nucleotides of the RNA showing. And what you've got out here is transfer RNA. They're transferring amino acids. A particular amino acid will have a particular anticodon. And again, it's A goes with T, G goes with C, and you get the one that fits what's here. That dictates what amino acid goes in that place. And then the ribosomes move down to the next codon, which is 3 nucleotides. The proper transfer RNA drops in the next amino acid, it links, and suddenly you get yourself a growing protein chain. Okay, this is the decoding process. It's the ribosome going in groups of 3 amino acids. And every time it sees 3, it calls in the correct tRNA, which brings the correct amino acid. And that means that whatever was the sequence coded in the DNA that was transferred to the RNA is now getting decoded into a sequence of amino acids. And that linkage with the sequence, it will then fold. And until recently, you needed an X-ray crystallographer in order to know what shape your protein is going to be. And now there's AI programs that you can run from your home computer. And you can put in your amino acid sequence, and it will actually show you a fairly good representation of what the shape of your protein is. So it's quite amazing. Now, the thing is that I think you can get the idea that to figure out triplets of nucleotides, to put that together with amino acids was, to break this code was difficult. It was as difficult a code to break as any of the codes out there that have been broken. And so that's why these people got the Nobel prize for working this whole thing out. The interesting thing about DNA and this code that we've gone through and how it's encrypted and unencrypted is that it's universal. Is that all organisms on Earth are made out of either DNA or RNA. So nobody can view themselves as being alone and detached from the other living organisms on Earth. Now, because we're all using the same coding and decoding and four-letter alphabet, it does mean that some of the small ones can parasitize the larger ones because the machinery is quite similar, okay? And it gives an opportunity for genetic material to be exchanged between the little ones and the big ones. And so what happened is that after the structure of DNA was found in the 50s, it sort of became etched in stone that the biological flow of information was linear, that it was DNA to RNA to protein. And this is the central dogma. And that some virologists, Howard Tiemann and David Baltimore, they were working on some RNA viruses, but the RNA virus couldn't interact with the messenger RNA because it was a negative strand and had to make a positive strand, but there was no enzyme to do that. And then David Baltimore and Alice Wong found the enzyme that would do that. So it means the virus can get into your cells, and if your cell doesn't have the complete toolkit for making the virus replicate and make more copies of itself, it simply brings in its own Allen wrench. That's it. And then Howard and David found some viruses in chickens and in mice, and they put them into cells where they turned the ability of the cell genome to make DNA off, and the virus wouldn't replicate. It wouldn't. And when they put them in the cells with the ability to replicate the DNA that's on, the virus could replicate. So they said, we've got some viruses that have mandatory in their life cycle that they actually convert their RNA genome to DNA. And the scientific community said, no way. That can't happen, that's too far out. And besides which, there's no enzyme to do that. So they went about to work, and yes, they found the Allen wrench. The virus brings it in with them. It's a reverse transcriptase. And the only reason why it's called reverse is because it's going in the opposite direction of the central dogma, which means the central dogma doesn't hold any longer, okay? So that if you've got the right enzyme and RNA can go to DNA and then back to RNA and then to protein. Yes, so somehow after telling Barbara McClintock that genes can't jump, they hadn't learned a whole lot about new information coming out, and they basically said no. And then, and just like with Barbara, there are jumping genes, and there are viruses that, retroviruses. So some of these viruses then can, when they're infecting an organism, get into the germline. And if they're in the germline and they stay there, they can go for generations. And when I say generations, I'm talking 500 million years. All right. And then we have something like COVID, where you can get horizontal transmission, where somebody infected can then spread it to the nearby, so that's person. Okay, so what they found after Barbara said there's jumping genes and everybody said no, was we had the time when molecular biology took off and people were using enzymes and kits and bacteria and all the rest of it. And they found out that bacteria have two types of DNA. They've got a plasmid and a main genome. Oh, that's like us, a main genome and a mitochondrion. And what happened was antibiotic resistant genes to pop out into the main genome. And then the other thing too is that if this bacteria didn't have the plasmid, and say it had antibiotic resistance associated with it, could get itself picked up by other organisms in its environment. So you've got this exchange between organisms and you've got this internal exchange as well. And then sometimes these bacteria, they're single cell organisms, but they can be infected by viruses themselves. They're called bacteriophage. And the bacteriophage can get in there and deliver DNA or take DNA, and then when the bacteriophage infects an adjacent organism, it can spread antibiotic resistance to a bacterium that didn't have it. So there's all kinds of exchange, and this is now known. Now, in oral bacteria, and I put this up because this whole rewriting of the Book of Life, this is not a history lesson, in that I'm not talking about everything that happened in the past. Things are happening right this very moment. I can get into that in a minute. But things are happening recently because this giant mobile genomic element that was found in oral bacteria is recent. The other thing I want to mention is that in the lab, you can have a lot of standard operating procedures, and you can find that you can culture one way and not find a certain trait. But if you grow the organism up a different way, the trait is there. So you have to be careful about standard operating procedures. And then, remember I said that we had our genomes in the nucleus, and then we had the mitochondrial DNA up there, and we're getting the idea that DNA loves to hold hands with another molecule of DNA. Well, they found that under stress, the mitochondrial DNA can fragment, and it can go out into the cytoplasm and it can get picked up in the nucleus. Now, is it going to be interrupting a gene that you really want to be functioning? Good. So they do walk around. Apparently, host stress, one of them is inbreeding. And that takes me back to Barbara Clintock and her corn. It was inbred corn. And it seemed as though that then, if those ears of corn thought that they were being stressed, what did they do? They created an awful lot of variety. And now one of the stresses here is that if we run short of the pool of deoxyribonucleotides, and there's only ribonucleotides available, that the mitochondria will go from the RNA pool and not the DNA pool. And the RNA pool makes the mitochondrial DNA fragile and is more likely to fragment. Now, here's jawed vertebrates that we're in that group. Humans are jawed vertebrates, and it's the B cells in us are the only cells in the human body that know how to make antibodies. They're the only ones programmed for it. And the antibody is a Y-shaped molecule, and it's got the combining sites, and both of them are identical in shape. And They're part of the fur that's out there on the surface of the B cell, and it can pick up anything that's floating around behind it. And here's the math problem. Okay, so the Human Genome Project figured out that in our 46 chromosomes, we code for about 20,000 genes. Okay, we make approximately 1 trillion different combining sites. Okay, now these Y-shaped molecules are actually 4 chains, 2 heavy chains and 2 light chains. And so we've got the heavy chains down to the bottom here, the heavy chain down to the bottom. The light chains are just here. So the arms have both heavy and light, and the stem is just heavy. Okay. So here's a question. How can so many differently shaped proteins be made from so few genes? Let's find out. Okay, so in the bone marrow, and that is in the absence of antigen. Okay, so if you've got a COVID infection and it's coming in and the surface antigen is triangular shaped, there is nothing in the human body that puts an order in to the bone marrow that says, anti-triangle, please, you know, stat, and it doesn't work that way. What happens is that there's differentiation of cells from pluripotent cells. Then you start making a progenitor B cell, which goes through a maturation process. Goes through a maturation process, and you get B cells. T cells also start out in the bone marrow, and they go up and finish maturation in the thymus, which is why they're called T cells. Now, there's different developmental stages of the B cell. When you talk about a B cell, there isn't really such a thing as a B cell unless you define it. So that it can be a naive B cell, that is before it's seen antigen. You can see an activated B cell, that's after it's coupled with antigen, and then it gets all worked up and goes into division and all that. You've got a B memory cell that's got an antibody on the surface that's just got the correct shape. That antigen comes in again, and it's going to lock onto it really quickly and tightly. And then you've got B regulatory cells, which can actually dampen down a response. And they're good in the cases of preventing autoimmunity. And then you've got the T cells, and you've got helper cells, cytotoxic cells. The cytotoxic cells are the assassins, they're serial killers. And then you've got T memory cells, and then you've got T reg cells, which just the other day, people got the Nobel Prize for regulatory T cells. So this is our human adaptive system, which is the B and T cells together. Okay, so they went to the progenitor B cells and went to look for the genes for the heavy chain and the light chain. They couldn't find any. There weren't any genes there. And so what Tamagawa did was he realized he found 3 different pools of gene segments, variable, diversity, and joining. And then we've got the constant region is the stem. So all he found was gene segments. And so really this is the first flat pack that, you know, some assembly required, because you have to build, and I'm talking just not for demonstration purposes, only the heavy chain on chromosome 14. You've got the same thing going on chromosome 2 for the light chain, chromosome 15 for the light chain, and chromosome 7 for the T cell receptors do this fragment building exercise as well. And so what happens is you need to put V, D, and J together so that it makes a complete DNA gene that can make an mRNA that can go to the endoplasmic reticulum, get the ribosomes to lock on, get it in frame for those, because it's going to be reading 3 by 3 by 3 by 3 to get the correct amino acid in there and the correct folding, so that you can get your heavy chain, your light chain, 2 identical heavies, 2 identical lights, makes the 4-chain antibody. Okay. Right. The thing about it is that you need to cut your DNA chromosomes up into fragments. Okay. And so it turns out that about 500 million years ago, a DNA transposon got into our human ancestors' genes and has been there for 500 million years. And it's the reason why we today have an adaptive immune system. It's why we can fight off all these pathogens that want to infect us and make us unwell. And it comes with 2 parts. One is a pair of scissors, which is here. The RAG1 and RAG2 proteins are the scissors. And actually gives a dotted line that's a cut here. And here's the dotted lines here. These are combination signal sequences composed of heptamer, nonomer, nonomer, heptamer, heptamer, nonomer, there we go. And what happens is that this is where the RAGs cut, is right here by the J and the D first. This RSS makes a signal joint, perfect joining, not a single nucleotide lost, perfect. And then the whole thing is sent to garbage disposal. It's completely broken down and it's gone. This is DNA that you were born with, it's gone. Then V goes to D, again, cut here, join the V to the D, all the RSS, which is all this DNA that you were born with, is gone. Okay? And then you get the lovely, lovely joining here, not a single nucleotide lost. It's brilliant. And then you've got the V to the D and the V to the D to the D to the J, and you've got all these 2 coding joints, and nothing could be sloppier. Nothing. We're going to get into that in a second. Okay, this is, say, what went on in one B cell. The next door B cell chose a different D that went to a different J. Different DNA in the middle got taken out and destroyed. A different V went to the DJ, and different DNA in the middle got taken out. That means that the B cells are not genetically identical. That means that all of your somatic cells are not genetically identical to each other. So even if you had an identical twin, you wouldn't be genetically identical to your identical twin because you're not genetically identical to yourself. And in the bone marrow, all this cutting and pasting and all that is, and so these are chromosomes getting cut to fragments, right? It's happening right now in the bone marrow to all of us. Now this is just to let you know when I say that the junctions are messy, is that when the D and the J, when it got cut, an enzyme comes in to break this hairpin that gets formed, but it doesn't do it symmetrically. So something that was on one strand gets flipped up to the other strand. Something that was on one strand gets flipped down. You've got a single strand overhang. Oh, single strand. Oh, exonucleases love those. They're just like a Pac-Man that just shoot them on. It's gone. Okay. And then sometimes good old DNA polymerase can fill things in using the other one strand as a template for the other. A go to T, G goes to C. But my all-time favorite enzyme, terminal deoxyribonucleotide nucleotide transferase, it's an enzyme that goes to the DNA pool and randomly, without use of a template, forget about the AT and the GC stuff. No, it just fills in, okay? And in one cell, it's filling in with one sequence, the other cell, it's filling in with another sequence, and all 10 to the 12 cells are filling in with different sequences. And that's how they're not genetically the same. Okay, so after looking at the corn stuff and the human stuff, and the retroviruses, the people that organize and categorize did a wonderful job saying that we've actually got two classes of transposable elements. The copy and paste ones, these are the retroviruses that were RNA, go through DNA, and then have to go back to RNA to protein again. They copy and paste. That's the RNA is copying into DNA and then has to copy back out RNA. The things that Barbara McClintock was looking at was cut and paste. Hers got cut out of a segment of the chromosome and got inserted elsewhere. And for the retroviruses, there's a pattern. The retroviruses have a group antigen, a polymerase, and a non-envelope gene surrounded by long-term repeats. That is what is found consistently. And there's a retrotransposon doesn't have the envelope gene. It does have the long-term repeats, gag and fall. And then there's all kinds of various other versions of things going on. But these sequences are what gave the people doing the analysis of the Human Genome Project a headache. Because if you've got an open reading frame, you've got to start, you've got to stop, you've got amino acids, everything, you can program your computer with search terms and you can find it. And then you start, and some of these things, like what Barbara found with one of hers, is that the one that was doing the moving actually couldn't move itself. It needed a neighboring mobile transposable element to make the transposase that would cut it out and make it move. So how, this is why the Human Genome Project really has been difficult. And so here we are, this is where we've got the DNA encoding with genes with functions at 3%, which everybody I think thought was going to be like 90%. Okay. And look at all the other categories. Here we've got endogenous retroviruses. This is like 8%. These are viruses that got into us. The most recent ones might be 6 million years ago. Some of them are way more million years ago than that. Okay, and then you've got DNA transposons. So yes, we've still got genes that can move around. And things that don't have the long-term repeat. And here we've got other non-coding. I mean, the enormity of figuring out what's been going on, it's truly amazing how much information has been obtained from this Human Genome Project. Launched in 1990, completed in 2003. And, you know, over half of it is made out of repetitive DNA. They've got 20,000 genes when I expected about 100,000. Okay. Yeah, about 8% has got these retroviruses, infections that occurred millions of years ago. Now, are all of these jumping genes, and especially the ones that are mutated and can't jump any longer, they're still there, they're still getting replicated, they're still going on generationally, but they can't replicate anymore. Just because they're not coding anymore because they're mutated, it doesn't mean they're not functional. And because some of them actually have been, well, they made another agreement with the cell saying we'll act as a coding regulatory sequence. We can help turn on the transcription of this area, we can help turn it off. And so we can find depressors, we can find accelerators, and you know, this kind of thing. So even though they don't actually make proteins and they don't make viruses, they don't make an infectious particle, it doesn't mean they're without function. So actually, yes, these endogenous retroviruses are part of a dynamic system. Yeah, and it continues to reshape our DNA by these genetic, these mobile elements. And if we were to get an infection today, either by HIV or a human T lymphotropic virus, if they're interacting with all of these endogenous viruses, even though they're mutated. And for example, they can make HIV can come in with its own envelope virus, envelope protein. And then if it wakes up, because HIV has to go through the provirus. But guess what? Here's a 6 million year old retrovirus, provirus, that's a next door neighbor now. So what happens is that the virus that you got 2 days ago is waking up one that you got 6 million years ago. Okay? And so what happens is that this is now making an envelope protein, and now you've got this virus trying to assemble, and it's got two choices now. What envelope protein can I use? And so sometimes it makes a mixture and puts one out with the retrovirus envelope protein on it. But the envelope protein are the keys that a virus uses to unlock the host cell it's going to infect. So now you've put a different set of keys on the outside of the virus. It can open doors that it couldn't have opened before. This one can open doors that this one can't. So now you've got a host range change. And then just a last example of a modern virus that you can get like today, which is Epstein-Barr virus, and it's a herpes virus. It can infect B cells, and when it gets into the B cells, the enveloped protein of the 6 million year old provirus gets made. And what this protein can do is it can sit on the outside of the B cell and get the B cell to hold hands with a T cell that it never before would have held hands with, because when they normally hold hands, there's a lot of shape specificity. It's like trying to click Lego bricks together. Got to be right there, spacing has to be right, click, okay? And that's the normal way of a T helper cell helping out a B cell and it's all controlled, and it's all based on shape. And here, it doesn't care about the shape of the antigen that this B cell has, because this protein binds not in the groove, not the normal combining site on the inside of the protein. It's binding on the outside surface. And that's why it's called a super antigen. It's because it's binding on the outside surface, and it can bind on so many more outside surfaces than when it goes through the normal recognition process. So you get binding between molecules and cells that shouldn't actually be saying hello to each other. And so you can get inflammatory diseases and autoimmune conditions. So from jumping genes, there are examples of diseases that it's felt that they're involved with. The benefits, they generate genome diversity. Oh yeah, we're good at that. We have an adaptive immune system in us jawed vertebrates. They can give us gene expression regulation, and people are now studying these endogenous retroviruses in more detail to see their potential as therapeutic agents. So these are my conclusions, but this is where the discussion happens with you, because you may have more conclusions than I've got here. Biological systems aren't always economical, and I can tell you that those B cells that are making the bone marrow, 90% of them die before they get out into the tissue. So yeah, they encourage errors. A lot of times, something I know for antibodies is that there's a stage in lymph node called hypermutation. And this is where the stars of the class, the gold medal winners, are the ones that make the most mistakes, okay? And yeah, some enzymes do have erasers at the end of them. If they make a mistake, you fix it. And these enzymes for somatic hypermutation, you make a mistake, you don't fix it. It's hallelujah, go on. Biological systems encourage diversity, just like the kernels of corn. We're all supposed to be colorless, and they went from full color to no color and everything between. They're not static. The books of life are constantly being rewritten. Now, here's the other thing. When we find, it is exciting to find explanations for the exceptions to the rule. So that if you study the exceptions, that might be the route into finding a new rule. And this is where the new discoveries are going to be. So basically, an open mind can happen as long as you check out that all the study was done correctly. Then it's time to investigate. I want to thank you very much for your attention.
Thank you, Catherine. We'll take a couple of minutes now just to have a bit of a break, and we'll start going on discussion in a moment. Thank you. Yeah. Okay, everybody, we're just going to get going again. I'm just going to add a tiny bit. We've had such a great explanation of how DNA makes RNA makes proteins today. I thought I'd add a little tiny bit that might interest people. I'm going to use the computer cursor here so that maybe the people online can see. Okay, on the dead spot again. So you might see I'm pointing at the sugar molecule. That's the ribose molecule, or rather the deoxyribose. The bit I'm pointing to, the reason it's bare, there's no oxygen on it. That's where the oxygen would be in ribose. What that does, it can then reach this, which is a normal size bond, not a really long one. That's an exaggerated one. it can reach down in ribose and attack that phosphate, and that's how it cuts itself. It's that mechanism, which is the one by which RNA, which is out and about in the cell making proteins, gets cut up automatically. It self-cuts after a very relatively short time, and that's why we then don't go and overproduce proteins, which would be really dangerous. So it's one of the most interesting bits of very, very simple chemistry. One little thing different. Deoxyribose, DNA, much more intact, stable for a very long time, can't cut itself. RNA cuts itself up, which is one of the most amazing things. Otherwise, we might end up with all sorts of different biology. The rate of break is all down to, it's going to be the pH and solubility, and it's going to be the solution in the cell. Because of course, normally it's doing jobs, but if it's not making something, then it would be prone to cut itself up. It's a small number of hours. I remember being told it was 60 hours, which is the average half-life for self-cutting, but it's not much. That's really useful. Anyway, I've gone back, but Kathy, do move around if you want to in any of your questions. So I'm going to take questions from people online as well. Okay, but I'm going to ask one of my own first, host privilege, just to ask, do chromosomes have reasons why they would break naturally? You're talking about fragmentation of DNA and chromosomes. Are there ways in which they would naturally fragment? And is this something that happens on nuclear DNA, or does that only happen in mitochondrial DNA?
Yes, during transcription, the DNA double helix.
Sorry.
Am I in the right place? Okay. The DNA has to unwind in order to let the polymerases in and the enzymes in and have the transcription take place. This unwinding and getting sort of the two strands separated from each other makes them more vulnerable to attack by enzymes and being cut. And also, if a DNA wanted to insert itself, if it is going to elbow its way in, it's not completely random. The easiest place to stick itself is a place that's already opened up. And then it can get in. There's things like sequence homology, which helps two DNAs hold hands. But a lot of this DNA jumping doesn't depend on sequence homology. It's a transposase that's it cuts DNA, double-stranded DNA, meaning it's going to fragment your chromosomes. And the transposase makes the opening, the gene goes in, you've got ligase, which is the glue that seals, and then there you go. And that's why it's in that place and not anywhere else. Especially if you get too really coiled, no, they're not going to get in there. But there are some conditions apparently where even when the DNA is circling on histones, that it doesn't do it tightly. And that also makes the DNA vulnerable.
Thank you. Anyone in the room? Okay.
Hi. Thank you for the talk. It was great. My question is related to the HIV infection. You mentioned that. Can you move back? Yeah. You mentioned that the HIV virus infects the cell and then waking up the dormant virus. How does that happen?
Transcriptases, because the dormant virus, especially this K grouping of dormant human endogenous retroviruses, the envelope gene is intact. It's a complete open reading frame, can make a perfect protein. It's usually repressed. Either the DNA can have methyl groups on it or something. It's normally repressed. But when the HIV comes in and provides transcriptases, it overrides all the controls that are on that gene. And suddenly you're now getting the transcripts off of the envelope gene, which gets translated into the protein, and the protein apparently has very, very sticky for the HIV matrix protein. Yes, and loves to get in there during assembly of the HIV and the matrix lines up. Oh, it's like a magnet. The envelope protein gets in there.
No wonder it's so difficult. So somebody online is asking, what is the relation of all this method of jumping genes to epigenetics?
Epigenetics, okay, is genetics is the DNA sequence, okay, and epigenetics is sort of a bit of, I don't want to make it sound trivial, but it's ornamentation on the DNA sequence. It's not a change of the DNA sequence. It's like methyl groups or something that is decorating the DNA. Okay, and so the jumping genes, even in this example with the HIV, it can be potentially overriding some of the regulatory signals, and epigenetics is part of the regulatory system for the expression of genes, and so it can interrupt the regulatory system.
I remember from one of your earlier slides, you were saying that the jumping genes were being controlled by their host.
Yes.
And I was fascinated by this because I can't decide, I want my genes to jump today. So
Right.
What's this level of control, and why do we think it's not random?
And that's the line that Barbara McClintock put into one of her early papers in the 1950s, and that's what got everybody worked up. Yes, so it's good spotting. And that it's the host is always in an environment, so you can't take the host out of an environment, okay? And when the cell is under stress, I don't know the sensory mechanism, but it tends to know that the cell is under stress. Now, we're going to call a stressed environment negative, okay? Not harmonious, it's stressful. And somehow the chromosome is detecting that it could die, okay? That the cell that it's residing in could die, and takes action and starts moving things around, which creates diversity. And the variability tends to mean that within that environment, one of the products of the diversity will be able to survive in this situation. So it's viewed as the organism taking defensive action against a hostile environment.
But it's just a cell in the organism doing that. It's not the brain saying you should do this.
It's not exactly.
And presumably the things that affect the chemical makeup of the cell, so heat does, which is one point, but also the pH, the acidity or basicity of the cell, that can then change what reactions happen, which could change whether or not the cell is detecting that stress level and starting to move DNA around. So that could be the chemical mechanism why that works.
Thank you, great question.
We've got one online. If identical twins are not identical, why do they look the same? I'm clearly missing something basic here.
That's because all the other somatic cells are identical. And what I was talking about are the cells in the liquid tissue. which is the blood. For non.
So the kind of structural cells, skin and hairs and all those kind of things, and tendons and muscles, they're more identical, but it's the ones that make up the immune system that change.
Yeah.
So would that explain why you could get one twin might be very susceptible to common colds and another one not?
Absolutely.
Okay, another one online. Do jumping genes relate to how we always seem to get infections and other debilities when we're under a lot of stress?
The short answer is stress has a lot of outcomes, and jumping genes is one of them. And depending on what genes are jumping, and there's going to be so many outcomes from jumping genes, because when they jump, they could potentially jump into the middle of a functional gene that the cell really wants for survival. You know, it's like ripping page 3 out of a book and shoving it in chapter 17 or something.
Or mixing up the code for a computer program.
Oh, there you go. Yes. So, it changes the story when the genes move around. That's the creation of diversity. But the diversity also promotes life, because you've got more chance of something being able to survive in that environment.
So you mentioned on one of your last slides, you mentioned the kind of relationship to cancer. Does this start to explain why some cancers appear to arise out of nothing in perfectly healthy people? Could this be a mechanism?
And it may appear that it's coming out of nothing, yes, but it could be because of a virus infection. And then suddenly, because they even said in COVID that they were looking for this K category of endogenous retrovirus proteins, and they found them being expressed in people with COVID. So when you've got, an active infection, you can be waking up things that normally are regulated, quiet, shut down. You never hear from them for 6 million years, and you've got them working.
And this also links the fact that a very large proportion of people who get type 1 diabetes do so after a fairly large infection.
Yes.
It's a significant proportion.
And it's this envelope gene from this 1K18. The fact that it acts as a super antigen, meaning that it's not restricted to fitting into the combining sites that have been made and sometimes selected for a special purpose. But by binding to the outside, there's a lot of proteins that have similar structures on the outside, so that you've got one protein that can bind and bring together many different cells. And you're activating a lot of T cells, and some of these T cells, and T cells can become cancerous. And you can start activating cells that suddenly, so if you have a B cell that actually has specificity for a self-antigen, not all of them get killed. They're supposed to get killed off in the bone marrow. Some of them get through. So you've got an anti-self B cell. Normally it doesn't have a B cell to talk to, so it just dies. But now, if you've got a super antigen to activate all these T cells, and these T cells and this anti-self B cell can get stuck together just by this super antigen binding on the outside, suddenly you've got the activation of an anti-self B cell that can make anti-self antibodies, and now that's autoimmune disease.
There's an absolutely great question online. How do we know that some of the genes are 500 million years old?
I'm leaving that to the evolutionary biologist. And apparently, there's the division of and creation of the jawed vertebrates. that the evolutionary biologists are saying happened 500 million years ago, and that we humans have evolved from the beginning of the jawed vertebrates, because we are a jawed vertebrate.
So this is pushing together, presumably, the bone, the fossils and the bone records that they can discover, with the genes now known to be in those classes of species. So it's all about understanding the tree of how life evolved into different branches.
That's right.
Okay, thank you. So you said at one stage that bacteriophages effectively can push antibiotic resistance into bacteria. Do we understand that well enough yet to be able to try and find a way to stop that happening? Because that's clearly very important.
They mainly pull it out of bacteria and make it part of their chromosome. It was already in the bacteria with the, say, ampicillin resistance gene is sitting there, and it's mobile. And then when the phage comes in, the mobile gene pops into the phage. The phage does its replication thing, makes many copies of itself, get out of the bacteria, and now they go and infect an uninfected neighbor, but they're now carrying out, they're extra long. They're now carrying an ampicillin resistance gene. And so now a host that previously wasn't ampicillin resistant is.
Thank you. Catch my eye if you want to ask anything. I can keep looking online as well. You were talking about mitochondria which can have the DNA kind of expelled from them. What happens to a mitochondrion if that occurs?
I haven't done a lot of reading on that topic, so maybe somebody in the audience that knows more than I know. I do know that when a circular double-stranded DNA is fragmented, its original function is no longer there. However, if that was to happen in 1 or 2 or however many, when the cell has got several thousand, it may not impact the health of the cell. If the central store of deoxyribonucleotides is depleted, and it affects all of them, and they're all pulling in RNA and making DNA-RNA hybrids, and that's all an unstable structure, and they fragment and can't do their energy production, which is what they're there for. I would say the longevity of that cell is in question.
The last question I had was, you were talking about how little of our genome in humans is actually used effectively. It's quite a lot that appears to have no function or is nonsense, and there's other bits which we now know have function, but we thought not to. The term nonsense DNA has been used an awful lot when talking about DNA kind of in public. Where it really has no function, is this the mechanism by which it would have got in there, if you like? Could it be that much earlier on the Earth, the organisms had much smaller genomes and they have expanded over many generations because these little bits of extra stuff get put in?
I kind of liken our genome to a patchwork quilt. That's why I call it the title, that it's sort of, you know, getting edited by these viruses and these mobile units that come in can then lose mobility, that if they're around for several thousand years or million years, have undergone enough replications that they have got quite a few mutations and don't function anymore, and we're still carrying them, and they may not even have a regulatory function. So Yeah, we have the evolutionary biology that says that we keep changing. I'm not sure if they're getting bigger with time, or if we're shedding some and gaining some, or whatever. But the thing is that the genome is not static. And that, I think, is the greatest evolution of human thought. Because around Barbara McClintock's time in the 1940s, is that no, the chromosomes are all fixed. Okay, end of story, no discussion. Okay, and I think the evolution of human thought is that there's room for a lot of discussion when something new is found.
The only thing is, though, so our own genetic code is not entirely stable, it's tweaking constantly in this way. But if we're going to pass it to the next generation, that's got to go to our gametes, that's got to go to the cells which form the next organism. Presumably that's a little harder to do. I don't know how much. It wouldn't be easy to change the DNA of eggs or sperm, would it?
No, it's harder to do, but if some of these viruses can get into the gametes, then you've got your vertical transmission, and it can happen.
Interesting. Got one more online. Might jumping genes also be implicated when, for example, people getting glandular fever, don't they often develop ME afterwards? And maybe there are other similar examples.
Well, I think that when with glandular fever, yeah, you've got the virus infection, and that's everything. The virus would do one thing in one human, slightly different than another human. That's because every human is providing a different environment. And so that if there are outcomes, you know, in consultation with the clinician, then that it's one thing to look at. I think it's something that this kind of information is where basic research and clinical medicine really do start talking to each other.
And it may not be the most urgent thing in clinical medicine to work out why people get glandular fever and then ME. Possibly. There are other diseases which perhaps more lucrative to look at.
Right, but I
I'm a cynic.
if you really, because this is where, when I mentioned that some of these endogenous viruses are now becoming targets of new therapies, this is where you've crossed over and you're having a conversation with clinical medicine who are getting an understanding of what's going on at the molecular level, and then can they do something that either interferes with an outcome that we don't want, or helps promote an outcome that you do want.
Yeah, probably a side question, but in the case of cancer, obviously the effects of the T cell are unable to defeat it. Is this something that will come either genetically or be able to tell the RNA that these are the bad guys?
Right, so some of the T cells have lost the ability to recognize a dangerous situation, okay? And they need to be wakened up to the threat that's right in front of them. And that's why some of these vaccines that are coming out are waking up T cells to the threat. The T cells are already there, they're just not seeing it. An antigen is being given, and they go, oh, okay, I see you. And then they go, oh, you're also on this cell over here. Oh, I didn't see you before. And now go and attack. So yes, there are current systems in place for waking up the immune system to threats that are actually, in fact, you know, they're not self, but the immune system wasn't seeing it as anti-self, and now they're working anti-self.
So bodies are a bit naive.
Well, it's hard sometimes to not allow the cell got blinded to the situation, and then to say, "Oh, hey guys, over here," and they go, "Oh, right, yes," and then they start doing their thing.
Lovely. I'm going to stop because we're pretty much out of our time and try and clear up time before I take Kathy and house for dinner. Thank you very, very much indeed. Before I do give you a last round of applause, just a reminder again, if you haven't joined the mailing list, then do. If you want to look on the website, you can see the next few messages. We've gone a bit biological at the beginning of the year this time, but there's a whole load of different stuff coming later, so you know, bear with it. I do try and get a variety over the year, but to be fair, I can't always directly work out. I can't decide where speakers are going to go. They have to tell me when they're free, so bear with it. Big thank you to Pharmacology. They are so nice to us here. They're not charging us for the room, which means that from your donations, I can afford the snacks and the drinks and all the technology which goes with putting this on in a nice, relaxing atmosphere. Do come again. If you've been coming online and you're in Oxford, try coming in person another time. And I'll see you, I think it's the 11th of November. There you go. What's this day? So good, easy one to remember. And then we are going with the earlier date in December, the 9th of December, our usual time, second Tuesday. I just have to cross my fingers and hope I can rush from interviews straight to it. But basically tonight, as always, these things don't work without a speaker. Kathy, thank you very much indeed for a very clear explanation.